This video tutorial is about the artificial use of plant cloning. For many years, gardeners and growers have used vegetative propagation. The easiest way to create clones is through making cuttings. To make a cutting, a stem is cut between two leaf joints, or nodes, and the cut end of the stem is then placed in moist soil, and that's shown on the left-hand side of your slide. New roots will grow from the tissues of the stem, usually from the node, but they might actually grow from other parts of the buried stem as well. Some plants, such as the blackberry, will take root really easily, whereas other plants may need further treatment. So one thing you can do is to dip the cut stem in rooting hormone, which will stimulate root growth. It's sometimes also useful to wound or remove the bark from the cut end of the stem, as this encourages the plant to produce a callus. For those of you that aren't sure, a callus is a mass of undifferentiated totipotent cells. Now, this technique can be used to produce large numbers of plants really quickly. Cuttings can also be made very successfully from other parts of the plant. So, for example, on the right-hand side of your slide is an image of root cuttings. And this is just where you take a section of root and bury that just below the soil surface, and it will produce new shoots. You could also do leaf cuttings. So you'd take a leaf and place it in moist soil, and then the leaf would develop new stems and new roots. Now, the problem is that large-scale cloning by taking cuttings can be really time-consuming and take a lot of space. Also, some plants don't respond well to taking cuttings. As a result, many commercially grown houseplants are cloned instead using tissue culture techniques. Tissue culture is a series of techniques used to grow cells, as well as tissues and organs, from a small sample of cells or tissue. It's carried out on a nutrient medium under sterile conditions. Application of growth substances at the right time can encourage the cells in the growing tissue to differentiate. Tissue culture is widely used commercially to increase the number of plants in micropropagation. So micropropagation involves taking a small piece of plant tissue, which is referred to as the explant, and using plant growth substances to encourage it to grow and develop into a whole new plant. Now, I'm just going to take you through the sort of steps of micropropagation. So first of all, a suitable plant material is selected and cut into smaller pieces, which are the explants. Now, they could be tiny pieces of leaf or stem or root or buds. Often meristem is used, as interestingly, meristem is always free from viral infection. The explants are sterilized using dilute bleach or alcohol, which is essential to kill any bacteria and fungi, not least because they would thrive in the conditions applied to help the plant grow well. The explants are placed on sterile growth medium, which is usually a kind of agar gel, which will contain the suitable nutrients required for growth. So, for example, glucose and amino acids and phosphates. The gel will also contain high concentrations of the plant growth substances auxin and cytokinins. This will stimulate the cells of each explant to divide by mitosis to produce a callus, which, as I mentioned before, is just a mass of undifferentiated totipotent cells. Once a callus has formed, it is divided up to produce a large number of small clumps of undifferentiated cells. And these small clumps of cells will then be stimulated to grow and divide and differentiate into different plant tissues. Now, this can be achieved by moving the cells to different growth media. Each media can contain a different ratio of auxin and cytokinin. So, for example, the first media might contain a ratio of 100 auxin to 1 cytokinin, and that would stimulate roots to form. And that's shown in the top of the diagram on the right-hand side. A second media might contain a ratio of sort of 4 auxin to 1 cytokinin, and that would stimulate shoots to form instead. Once tiny plantlets have formed, these will then be transferred to a greenhouse to be grown in compost or soil and sort of acclimatized to just normal growing conditions. Now, there are several advantages and disadvantages of artificial cloning in this way, so through tissue culture or through taking cuttings. You need to be familiar with these arguments. So first of all, cloning is relatively rapid, so it's a really quite quick way for producing new plants compared with just growing them from seed. Secondly, cloning can be carried out where sexual reproduction is not possible. So some plants that have lost their ability to breed sexually can be produced in this way. For example, commercially grown bananas. Similarly, plants that are hard to grow from seed can be reproduced, for example, auxins for the horticultural industry. Thirdly, the plants selected will all be genetically identical to the parent plants. They will therefore display the same desirable characteristics such as high yield, resistance to a common pest or disease, or a particular colour of flower. Fourthly, if the original plant had an unusual combination of characteristics due to sort of selective breeding or genetic modification, then this combination can be retained without any risk of losing that combination through sexual reproduction. Fifthly, the final plants, or the sort of the new plants, are all going to be uniform in their phenotype, which makes them easier to grow and harvest. And finally, using the apical bud, so the meristem, as an explant for tissue culture is really good because it ensures that new plant is going to be virus-free. On the flip side, there are several disadvantages of artificial cloning. So firstly, tissue culture is really labour intensive. It's expensive to set up the facilities to perform tissue culture successfully. All the cloned offspring are genetically identical and are therefore susceptible to the same pests or disease. So crops grown in monocultures are going to allow rapid spread of disease or pests between the closely planted crop plants. And finally, there is no genetic variation except that introduced by mutation.